Hello everyone, welcome to our food fermentation assignment. Our group did wine fermentation, and the members of our group are me, Jeffrey Bauer, Jacob Atuahene, and Julian Kobos. Wine production is really old. It's been around for a long time. The number of ways that you can produce wine are really diverse, but for the purpose of this presentation, we're going to talk about two specific methods of wine production, sweet and dry. Other things that will be covered in our presentation are one very important microbial organism that is crucial for wine production. We're going to talk about the growth factors of this microbe and how it relates to the production of wine and how it impacts characteristics of wine. We're also going to give you an overview of modern wine production methods, and we're going to talk about common modes of wine spoilage. Saccharomyces cerevisiae, or S. cerevisiae, is going to be the major topic of discussion for this wine production presentation. S. cerevisiae is the most commonly used microbe for wine fermentation, and in this project presentation we're going to give you a breakdown of its characteristics and qualities, as well as its a taxonomic description of this microbe. So, S. cerevisiae, also known as baker's yeast, is the most associated microbe with wine production. And it's a yeast, and yeasts are the microbes that are responsible for fermenting grape juice into wine. S. cerevisiae is a single-celled eukaryotic organism. S. cerevisiae are ovoid-shaped or spherical, and they're ascomycota, which means that they're sac fungi. And S. cerevisiae belong to a taxonomic group known as ascomycetes, which can produce sexually or asexually, but S. cerevisiae actually reproduce through an asexual process known as budding. The taxonomic breakdown of S. cerevisiae is that it belongs to the kingdom fungi within the division Ascomycota, and the genus is Saccharomyces, and of course the species is Saccharomyces cerevisiae. To continue, S. cerevisiae, like I mentioned before, is the most common microbe associated with the fermentation of wine. And the reason is because there's certain favorable characteristics that this yeast has that makes it really, really good for wine fermentation. So S. cerevisiae has the capacity to withstand high alcohol environments relative to other microbes. And this is important for obvious reasons, because wine contains alcohol, and the microbes that are used for its production need to be able to survive in that environment. Also, S. cerevisiae can withstand higher sulfur dioxide environments. Sulfur dioxide is a common additive that's used in wine production, and it's designed to kill undesirable yeasts and bacteria. Also, S. cerevisiae, it has a really strong and predictable fermentation capabilities that makes it really easy and favorable for wine producers to use. Next, I'm going to hand it over to Jacob, and he's going to talk about growth and fermentation.
When it comes to growth and fermentation, there are factors that impact the microbe growth, Saccharomyces cerevisiae in particular. They can tolerate very warm environments, ranging from 77 to 86 degrees Fahrenheit. Yeast growth will be at its greatest around 6.0 pH If the pH gets too acidic, all fermentation could come to a halt. Oxygen is kind of like a double-edged sword. On one side, it is a good yeast nutrient which helps the cells build membranes that can tolerate ethanol as well as speed up their metabolic reactions. On the other side, if the microbe is not the one needed for fermentation, the final product could be spoiled wine. A 60 to 80% moisture level is the ideal range for the wine storage container. This keeps the seal or cork from drying out and cracking. Once the alcohol levels in the wine reach about 15%, the fermentation process usually stops, even if there is leftover sugar in the grape juice. After yeast is added to grape juice, the hexose sugar molecules get broken down from triose sugar molecules, then convert to carboxylic acid. The carboxylic acids is further metabolized to acetaldehyde, then finally to ethanol and carbon dioxide. Volatile and non-volatile compounds are also produced in the process, contributing to the fruity taste and floral scent of wine. In terms of spoilage, wild yeast and bacteria play their part in destroying the pleasant taste and aroma in wine, as well as cause discolorations like browning or mold. Like I stated earlier on, the undesirable microbes will also need oxygen in order to survive in the grape juice environment and can help encourage the growth of acetic acid bacteria and lactic acid bacteria. Some common spoilage organisms include Acetobacter pasteurinus, Zygosaccharomyces bailii, and species of Candida. In addition to the microbes, improper storage practices and improper cleaning and sanitation can also mess things up. So excluding as much oxygen as possible and the appropriate usage of sulfur dioxide are good safety measures for the quality assurance of stored wines.
The two methods that we compared were sweet wine and dry wine production. I'll touch more on those later, but as far as the general manufacturing practice in wine, there are two phases. The first phase is the harvesting or the collection of the grapes or fruits to be transferred to the winery. This is a time-sensitive process. The time of harvest depends on the ripeness of the grapes, which accounts for their level of sweetness, acidity, and the tannins and other polyphenol compounds contained in the grapes. They can be either hand-picked, which requires the use of skilled laborers, or you can accomplish this through machine picking using a mechanical harvester. The second phase is vinification, which is the actual processing of grapes into wine. So the first step in vinification would be to destem and crush the grapes. So for red wines, the grapes are destemmed from their stalks using a crushing destemmer machine. And for white wines, the manufacturer may decide to allow some contact time between the skin and the juices after being freshly crushed. Otherwise, the skins are discarded as they're not used during the fermentation process in white wine production. The second step would be the preparation of the must, which is the juices that come from the grapes. So several additions or modifications need to be made depending on the climate or the environment that the grapes were grown in and what wine or wine style is being produced. So some examples of these additives could include sulfur dioxide for microbial control, sucrose added to supplement some of the sugars inside of the grapes. Carbonates or salts to raise the pH of the must. Tartaric acid is used to lower the pH. Either cultured yeasts using a yeast starter or naturally occurring yeast in the grape skin. And then nutrients to supplement the growth of the yeast, such as B vitamins. And then once the must is prepared, then the fermentation will actually take place typically inside of large steel vats or sometimes wood barrels. The glucose and the fructose in the grapes are, of course, converted into ethyl alcohol via yeast fermentation, and carbon dioxide as a byproduct. The sugar content in the must needs to be carefully accounted for in the methods that we chose to analyze. This is done using a density hydrometer, which measures the density of the must, and then using formulas will directly relate to the sugar content. Extraction is another process that takes place during fermentation. So this is the process of extracting color and tannins from the solid portion of the must, which would happen in red wine production. So the CO2 that's produced causes the skins and the other grape solids to float to the top of the must and kind of like a solid cap. And that needs to be constantly submerged or manually punched down into the must for adequate extraction. The next step is called racking. This can take place at several points throughout the entire process, but it's essentially the transferring of the juices from one container to another. The wine left after fermentation is called free-run juice, and racking essentially filters any solids or debris in the must from the free-run juice, and then is transferred to another vessel. Pressing is the step that occurs with any of the solids that are left behind after racking. So all of those are pressed at gradually increasing pressures, and the juices produced from that usually have more tannins and color and are often considered lower quality. Another fermentation, the second fermentation, is also called malolactic acid fermentation. This is done using bacterial inoculation instead of a yeast fermentation. So it allows other scent levels or additional levels of complexity to be incorporated into the wine, usually using strains of Lactobacillus, Leuconostoc, or Pedagoggus bacteria. Then the final alteration that needs to be made to the wine itself is blending. So wines of differing vineyard origins, grape varietals, or ages are blended together in order to create a wine that draws on the best qualities of each, and will usually represent the seasonality and brand style that is typical of the wine company. And then lastly, the wine has to mature. So maturation is the final step in vinification. Wines are analogous to fruits in that they essentially need to ripen for a while to balance out the flavors and the acidity level to be presentable to the consumer. So for sweet and dry wine production, essentially what these production methods boil down to are the amount of residual sugars left after fermentation has taken place. So for sweet wines, those will have a greater sugar concentration. So fermentation has been stopped prematurely in order to get a sweeter taste. For sweet wine, as far as microbial growth, high sugar concentration induces hyperosmotic stress onto the yeast cells, which makes it hard for them to grow and ferment continuously. It obviously makes the wine taste sweeter. Sugar content also impacts the perceived viscosity and density of the wine, so that has to be managed to make it palatable to the consumer. And then there isn't really an impact on the actual appearance as far as color. That comes from the phenolic compounds like tannins in the grapes, the varietal, the extraction process, and blending. And then as far as dry wine production goes, this is a more ideal environment. It's the tonicity is more productive for S. cerevisiae. And it's also influenced by other additives, like I mentioned before, like B vitamins and other things to help the yeast grow. The taste will be more bitter or dry, hence why it is called dry wine. Ethanol content and glycerol also impact the texture, so those in conjunction with sugar content need to be managed for the appropriate viscosity and density of the wine. And then, of course, the appearance is the same as in sweet wine. That's influenced by other intrinsic and extrinsic factors.